And the cytoplasm is going to be formed of the cytosol, which is simply the water and solutes. And the solutes include the proteins, salts, sugars. Also, it's going to have the cytoplasmic organelles. And the cytoplasmic organelles are going to be the metabolic machinery of the cell. Those are the machines inside the cells that will be carrying on all the metabolic activities of the cell. And I've got inclusions, which are like the granules of glycogen or pigments, lipid droplets, crystals, vacuoles, which are not directly participating in the metabolic activities of the cell. They are rather contents other than machines inside the cell. We classify the cytoplasmic organelles into two major groups. We have the membranous organelles, in which I have a structure similar to the cytoplasmic membrane, and non-membranous organelles, which won't have like any membrane in their structure. Cytoplasmic organelles include the mitochondria, the peroxisomes, lysosomes, the endoplasmic reticulum, and the Golgi apparatus. The non-membranous organelles, this includes the cytoskeleton of the cell, centrioles, and the ribosomes. Starting first with the most famous organelle in the cells, this is going to be the mitochondria. And as you see on here, the mitochondria is going to be formed of two membranes. I have an outer membrane and an inner membrane. The mitochondria will have their own DNA and RNA, which will allow them to replicate independently from the cell. So the mitochondria can increase their numbers inside the cell without actually having to divide the cell, without having to copy and produce the organelles from the cellular DNA. It has its own DNA, it has its own genetic information. The mitochondria in the cells are going to be responsible to produce ATP from the cellular respiration. So cellular respiration is a process in which we are going to be breaking down the glucose, for example, into water, carbon dioxide, and energy in the form of ATP. So the most critical steps of this process are going to be taking place inside the mitochondria. So we're going to call the mitochondria is the powerhouse of the cell, because this is where the energy is going to be produced in the cell. Another organelle that we're looking at today is a non-membranous organelle, which is the ribosomes. And the ribosomes are going to be granules containing protein and a type of RNA called the ribosomal RNA. Ribosomes would serve as a site for protein synthesis. This is where the proteins are going to be produced. And they are present in the cell in two forms. I have the free ribosomes and the membrane-bound ribosomes. Free ribosomes are scattered in the cytoplasm, like when you see on here. Those are my free ribosomes. They are scattered around in the cytoplasm. And I have the other type, which is the membrane-bound ribosomes, and those are going to be attached to the membrane of the rough endoplasmic reticulum. You see on here, this set of tubules that are located very close to the nucleus. This is an endoplasmic reticulum, and this type of endoplasmic reticulum gonna have ribosomes attached to its outer surface. So we're gonna call this is my rough endoplasmic reticulum. So what is the difference between the free ribosomes and the membrane-bound ribosomes, which are attached to the rough ER, the rough endoplasmic reticulum, in terms of function? They both are going to be responsible for the formation of proteins, but the free ribosomes are more concerned with the synthesis of soluble proteins that will remain as part of the internal structure of the cell, compared to the membrane-bound ribosomes are going to be responsible for the formation of the proteins that either would be incorporated into the plasma membranes or that would be exported to the outside of the cell. So again, two types of ribosomes. We have the free ribosomes responsible for the production of the soluble proteins, and the membrane-bound ribosomes, which are attached to the rough endoplasmic reticulum, that will be either producing the proteins that are incorporated within the cell membranes. Like what proteins that get incorporated with this plasma membrane, if you remember, like, for example, the channels. So those channels are produced by rough endoplasmic reticulum. The carriers, like the sodium-potassium ATPase pump that we've studied earlier. This is a protein carrier. So where does it come from? It gets produced from my membrane-bound ribosomes, which are attached to the rough endoplasmic reticulum. Carriers for the glucose, for example, like glucose transporters, those are proteins. The receptors, remember, some of the membrane proteins were acting as receptors. Some of the membrane proteins were acting as enzymes. Some of the membrane proteins were acting as junctions. Between the cells, like the lock in here. So those are proteins that get incorporated within the plasma membrane, and all of those are going to be produced from the membrane-bound ribosomes. Other than the incorporated proteins in the plasma membrane, we've got the proteins that will be released to the outside of the cell. If, for example, I'm producing a protein-based hormone, so this hormone is kept in a vesicle, and this vesicle is going to be transporting this hormone to the outside of the cell. So it gets exported to the outside of the cell. So this hormone, it's actually going to be produced from a membrane-bound ribosome, the ones that are attached to the surface of those flattened tubules called the rough endoplasmic reticulum. And again, the rough endoplasmic reticulum is the one that has ribosomes attached to its outer surface, like this. Moving on to another type of cell organelles. Those are the endoplasmic reticulum, and we have two types of endoplasmic reticulum. We have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. As you see on here, this is my smooth endoplasmic reticulum. I am formed of flattened tubules, and those tubules will have the same structure like the cell membrane. I'm a membranous organelle. And if you notice on the outer surface, they don't carry any ribosomes. This is why they are called smooth endoplasmic reticulum. Compared to the other one on here, you'll see I am formed of flattened tubules. They have the similar structure like my plasma membranes, and to the outer surface of it, we've got those ribosomes attached, the membrane-bound ribosomes. Membrane-bound ribosomes. Are attached to the outer surface of the rough endoplasmic reticulum. If we look into the rough endoplasmic reticulum, you will see that other than having the ribosomes attached to the external surface, it's gonna have a connection with the nuclear membrane, which is the membrane that surrounds the nucleus. It's connected directly to the nuclear membrane, the membrane of the nucleus. And it serves as the site for the proteins, again, that would be either integrated within the plasma membrane or that would be exported to the outside of the cell. As we've mentioned, the function of the membrane-bound ribosomes. So rough endoplasmic reticulum is simply going to have the same exact function as the ribosomes because they work together to conduct the same functions. Moving on to the smooth endoplasmic reticulum, it's also going to be formed of tubules arranged in a looping network. And the main function in here, the main functions are going to be determined according to their locations. So in the liver, it is going to carry different functions than what it's going to be performing. For example, in the intestinal cells, it's going to be different than the function in the muscle cells. So in the liver, the smooth endoplasmic reticulum is going to be concerned with the lipid and cholesterol metabolism. This is where I have the enzymes responsible for breaking down and metabolizing the lipids. Also, it's going to be responsible to break down the glycogen along with the kidneys. Other functions is going to be the site of the detoxification of the drugs, pesticides, and carcinogens. So this is where the metabolic activities of the liver cells is going to be taking place. This is the site inside the cells where those reactions are going to be taking place. In the intestinal cells, they would be responsible to allow the absorption, synthesis, and the transport of the fat. On the other hand, in the skeletal and cardiac muscle cells, this will serve as a site for storage, release of calcium. So generally speaking, what is the function of the smooth endoplasmic reticulum? Again, this is going to be determined according to where are you asking about. If you're asking about in the liver cells, remember what are the functions of the liver? Detoxifies the toxins, metabolize fat, break down the glycogen into glucose. So all those reactions are going to be actually taking place inside my smooth endoplasmic reticulum of the hepatocytes, of the liver cells. Something that is hard to deal with in terms of absorption. Unlike the sugar, unlike the amino acids, is going to be the fat. So I need a place inside my intestinal cells for me to carry the fat I'm absorbing and get it metabolized in a form that allows it to travel into my blood. So in order for me to metabolize the fat and transport it to the blood, it needs to be modified in the smooth endoplasmic reticulum of my intestinal cells. The third major location is going to be in the muscles, again, like the cardiac muscles and skeletal muscles, inside the muscle cells. As we're going to learn, we're going to have those sets of flattened tubules, smooth endoplasmic reticulum, waiting for any signal to arrive from outside. So once I get the signal to the inside of the cell, this endoplasmic reticulum gonna be releasing the stored calcium. And why do I need to release the stored calcium inside the skeletal muscle cells and cardiac muscle cells? For the cells to be able to contract, for the cells to shorten their length. What we're gonna simply do, we're gonna need to get a signal that tells the cell to contract. And this signal is gonna be reaching, as we're gonna learn in details, the smooth endoplasmic reticulum of the muscle cell for it to release the stored calcium out. And this is what actually induces the muscular contraction. So again, what are the different functions of the smooth endoplasmic reticulum? We've got in the liver, lipid, cholesterol metabolism, breaking down of the glycogen, detoxification of the drugs, pesticides, and carcinogens. Intestinal cells will be the absorption, synthesis, and the transport of fat. Skeletal muscles and cardiac muscle cells will be the storage and release of calcium to allow the muscular contraction. All right, we're going to discuss the muscular contraction and how the signal is going to be transported to the cell and how the contraction takes place. All this is going to be discussed in details later on in this course. Moving on to another membranous organelle. This is the Golgi apparatus, which is also formed of stacked, flattened membranous sacs. And those are going to be responsible to modify, concentrate, and package the proteins and lipids. The packaging that the Golgi apparatus you see on here going to be given to the certain protein is what's going to be determining the fate of the protein. What this means. So if you have a diamond ring, would you put this ring in a fancy box? Or would you put it in a plastic bag from Walmart? So what most likely gonna happen? What most likely gonna happen to this ring if you just put it in a bag from Walmart? What's gonna happen? Most likely to this ring. Would it be kept safe, or most likely you're gonna lose it? It's gonna get lost, right? All right, so what determines the fate of this ring is the type of packaging that you've given the ring, right? Where did you put it? And how is it packaged? You put it in a fancy box in a safe, yes. This makes sense. Like if you put it in a plastic bag from Walmart, it will find its way to the trash by one way or another. All right, so the same concept when we're dealing with proteins. So remember, where is the protein gets produced? It gets produced in the rough endoplasmic reticulum by the ribosomes attached to it, the membrane-bound ribosomes. Those proteins that you are going to be forming are going to be released in vesicles, and those vesicles will be fusing with the membranes of the Golgi apparatus, because the Golgi apparatus is going to be trimming, modifying the shape of the protein, finalizing the shape of the protein. Also, it's going to be given those proteins their final packaging that will be determining their fate. Like what? Like, for example, if I am a protein responsible for digestion, I am formed of I am a digestive enzyme in a macrophage or a white blood cell. So what would you like to do with this digestive enzyme? Would you like this digestive enzyme to be released outside of the cell to break down your cells, your own cells, or would you like to keep it in a vesicle inside your cell, waiting for another vesicle that contains the bacteria to come in for you to break down the bacteria? Remember when we mentioned the endocytosis, the types of endocytosis, we mentioned that engulfing a solid particle is called phagocytosis. So what you're going to do with this vesicle with the bacteria inside, what you're simply going to do is fuse the vesicle with another vesicle that contains digestive enzymes for the digestive enzymes to break down the bacteria. So you don't want to keep this bacteria forever living in your cell. So you're going to keep it in a vesicle inside your cytoplasm. How about if the proteins that you are producing is going to be a channel, like a protein channel that allows the sodium to pass into your cells? What you're going to do with this vesicle that contains the protein channel, you want it to become integrated within the phospholipid bilayer of the plasma membrane in order for this channel to be able to perform its function. So what did it allow this channel to become integrated within the plasma membrane and to not be kept in a vesicle inside the cytoplasm like for the digestive enzyme is a type of packaging it gets from the Golgi apparatus. What if I'm producing a hormone, a protein based hormone. Would you like me to keep this hormone inside my cell, never gets released to the outside? Or would you like this vesicle to fuse with the cell membrane, releasing its content to the outside for this hormone to be delivered to its target organs? What do you think? I would like this vesicle definitely to fuse with the plasma membrane to allow this hormone to be released to the outside of the cell. So we're looking at here at what did determine the fate of the proteins that get produced by the ribosomes within the tubules of the rough endoplasmic reticulum is the type of packaging it received in the Golgi apparatus. If it was a lysosome, it's gonna be kept, the vesicle gonna be kept containing the digestive enzymes within the cytoplasm. If I contain hormones, I give it a type of packaging that has the ability to fuse with the cell membrane and release the contents to the outside. If it's going to be integrated within the plasma membrane, I have to give it a type of vesicle that has the ability to stick to the plasma membrane and not to release the protein to the outside or to keep it on the inside. All right, does this make sense? Make sense? Questions? Any questions? All right, so moving on to another membranous organelle that we've got here is the lysosomes. And the lysosomes are a vesicle containing digestive enzymes. And they would be responsible to digest and break down. So what am I trying to break down? What would I be trying to break down? I will try to break down bacteria, ingested bacteria. I would like to break down viruses. If I got exposed to toxins, I would like to break down those toxins. I don't want to keep them. What else do you want to break down? If you've got organelles, which are non-functional. So organelles are not going to be conducting their functions anymore, like old ribosomes, for example. They are not producing any more proteins. Would you like to keep them forever in your cells, although they are non-functional? No, you're gonna simply be breaking them down to use their chemicals to build up other organelles, new organelles. If I want, for example, to produce glucose in the liver, I would like to break down the stored form of glucose, which is the glycogen. So I want to break down the glycogen into glucose. How would I break down the glycogen into glucose? By using the digestive enzymes that are located within my lysosomes. Lyso, to break down, lysis to break down, and om is a body, like oma is body. So those are the bodies responsible to break down, lysosomes. What else do you want to break down? In some cases, I have low blood calcium levels, so I want to break down the bones in order for me to release the calcium out in my blood. So if you are looking at the chemical structure of the bone, you will find that the bone tissue is formed of protein, and attached to the protein, we've got calcium. So what will I do to release this calcium from my bone to my blood? I will go simply and break down this protein. So now the calcium is free and it's gonna be released out in my blood, increasing my blood calcium level back to normal. So who's gonna be breaking down here those proteins to which the calcium is attached? Those are gonna be my lysosomes. So I'm breaking down the glycogen in the liver, breaking down the protein to which the calcium is attached to. Also, in some cases, I would use the lysosomes to commit suicide. So the cell is non-functional. I not carry on my metabolic activities as they should be. I have unuseful tissue. I have unuseful content. So the cell will decide, so I will get rid of myself. I will be using my lysosomes to release the digestive enzymes freely inside the cell. And this will allow the cell to break down itself. So we call this autolysis. Auto means self. And lysis is to break down. So autolysis means the cell gonna be breaking down itself. The tissue is gonna be breaking down itself. Destroy cells in injured here or non-useful tissue. We call this as autolysis. Also, it might be mediated by external factors. We're gonna get to this later on in AP2 on how can I induce autolysis and apoptosis. All right, so what organelles did we discuss so far? First, have seen the mitochondria. And remember, what is the function of the mitochondria? It's the powerhouse of the cell. It's going to be responsible to produce ATP in the cell. We've seen another type of organelles, a non-membranous organelle, which is the ribosomes. And remember, we've got two different types of ribosomes according to their attachments. Either they are located free in the cytoplasm, because those are by free ribosomes, or they are attached to the outer surface of the rough ER. Those are going to be my membrane-bound ribosomes. Another type of organelles that we've seen was the endoplasmic reticulum, which is formed of flattened tubules like this, membranous tubules. And remember, we do have two varieties of the endoplasmic reticulum. We have the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Remember, rough endoplasmic reticulum has ribosomes on its outer surface, which will allow it to produce the proteins that will either be integrated within the plasma membrane or that will get exported to the outside of the cell. Compared to the smooth ER, its function is going to be determined according to where is it located. So in the liver, it's going to be responsible for the metabolism of the fats, lipids, and detoxification of the toxins. In the small intestines, it's going to be responsible for the metabolism of the fat and its absorption. In the muscle cells, like the cardiac and skeletal muscle cells, it's going to be responsible for storing and releasing the calcium, which will allow the muscle cells to contract. We've seen another membranous organelle. This was my Golgi apparatus. And remember, the protein coming out from the rough endoplasmic reticulum will not be ready to function out of the rough endoplasmic reticulum. It needs to become concentrated, trimmed, modified, and packaged in the Golgi apparatus before its fate is determined. So remember, the packaging here is that the Golgi apparatus is going to be given to the protein is what determines its fate, whether it's going to be a vesicle that will keep it inside the cytoplasm, or will it the vesicle will fuse with the plasma membrane, releasing the contents, or it will be integrated within the plasma membrane for this protein to become an integral protein. We've seen another membranous organelle in which I have digestive enzymes. Those are vesicles containing digestive enzymes. Those are my lysosomes. And remember, the lysosomes are going to be responsible to break down. And what do I need to break down? Again, any non-functional organelle. I can break down the stored glycogen. I can break down the protein attached to which the calcium, is the calcium attached in my bone. I can even break down tissues by autolysis. Another membranous organelles that we're looking at today is going to be the peroxisome, and those are membranous sacs, similar to the lysosomes, but they have different content. So inside those sacs, we don't have digestive enzymes. We do have enzymes which are oxidases and catalases. And they are responsible to detoxify the free radicals, neutralize the free radicals, which are reactive chemicals that got produced with unpaired electrons, like H2O2, for example. H2O2, it's a harmful chemical. If it builds up in my cells, it's gonna cause the destruction of the cell. So what we're gonna do, we need something that will detoxify it and neutralize it into water and oxygen, which are much more chemically stable, unlike the free radicals, which can cause harm to the cells. And those free radicals gonna actually get formed all the time while the cell is carrying on its metabolic activities. So when I carry on the metabolic activities, some of those free radicals will get formed, but I need those peroxisomes to neutralize them for the cell to not get destroyed. All right, so our second question for today's in-class activity is what organelle is important for protein synthesis? Which one is going to be producing proteins? Ribosomes, the nucleus, mitochondria, or lysosomes? Which one is going to be responsible for the production of proteins again inside the cell? Let's see a few answers. Yes, here we go. Ribosomes. Ribosomes. Can you remind me how many types of ribosomes do we have? 2, either free ribosomes or membrane-bound ribosomes. We have only two types of ribosomes, 2 types of ribosomes, all right? 2 types of ribosomes, either free ribosomes or membrane-bound ribosomes. Moving on to another non-membranous organelle, and those are the cytoskeleton. And the cytoskeleton are series of rods that would be located throughout the cytosol, and they will form the skeleton of the cell. From its name, it's cytoskeleton. And those are microtubules, microfilaments, and intermediate filaments. So if we're looking on here at the first type of the cytoskeleton, we have the microfilaments, and those are formed of a protein called actin. Those filaments, as you see on here, they are occupying the whole cytosol. Actin subunits are forming those filaments of very small diameter, which is 7 nanometer. Another set of filaments that will be also occupying the cytoplasmic building of the skeleton of the cell will be the intermediate filaments, which will have a larger diameter compared to the previous ones. They will have a diameter of 10 nanometer, and unlike the microfilament, the protein subunits responsible for the formation of the intermediate filaments is going to be fibrous subunits, unlike the microfilaments. They were formed by actin subunits. A third type of filaments is going to be the microtubules, and the microtubules are going to be built of another protein subunits called the tubulin. Tubulin subunits are arranged in the form of tubules of a larger diameter compared to the previous two, which is around 25 nanometer. So started first with the microfilaments with 7 nanometer in diameter. Then we've moved to the intermediate filaments with 10 nanometer in diameter. And the third one is going to be the microtubules with 25 nanometer in diameter. Moving on to another organelles that we have in the cells. This is going to be the centrosome, and this is translated into the cell center. It's actually very close to the nucleus. Yeah, so correct. So you might see it different than in your textbook. Please stick to the information on the slide on here, please. Please stick to the information on the slide rather than in your textbook if you have stated differently. All right, please stick to the information on the slide. All right, so we're looking here at the centrosome, which is translated into cell center, which translates into cell center because it's going to be located near the nucleus. The centrosome is going to be responsible for the generation of microtubules. And those microtubules will be responsible for the organization of the mitotic spindle fibers. And each of those centrosomes, as you see on here, is going to be formed of two centrioles. And if you look closer to the two centrioles, you will see that they are formed of tubes of microtubules. So I'm building up the centrosome by microtubules. All right. Moving on to another type of organelles. This is going to be a part cytoplasmic membrane and part an internal core formed of the cytoskeleton. Those are the microvilli, which are finger-like extensions coming from the cytoplasmic membrane. And the main function here of having those microvilli is to increase the surface area for absorption. So they have an outer cytoplasmic membrane, and inside it's stiffened by the presence of actin filaments. All right, so again, what is the function here of my microvilli? Those finger-like projections coming out from the plasma membrane is to increase the surface area for absorption. What does this mean? So if I have two cells like this, one that has a smooth surface and the other one has microvilli, which one has better exposure to the outside? Number one or two? Which one has a better exposure to the outside? Number one. Why? You see, all this is exposed to the outside. Compared to a number two, this is what is exposed to the outside. So this one has much greater surface exposure. Number one with the microvilli has a much greater surface exposure. So it has higher capability for absorption. So all those sites are available for absorption. Compared to number two, much less absorption can be carried. So if it's like a string, if I pull on both sides here of the string, you see on here, if I make it flattened, this is how long it will be. If I pull the two sides away from one another, imagine it's going to be much longer than in number two. So where do I need microvilli to be present? Where do I need to have microvilli to increase the surface area for absorption? Where do you perform a lot of absorption? In your small intestine. And also in parts of the kidney tubules. In the kidney, if you remember, in some cases I would need to reabsorb molecules that have been released out in the filtrate. So I will need microvilli also in parts of the kidney tubules. So again, what are the microvilli? Those are finger-like projections that are coming out from the plasma membrane, and their main function is to increase the surface area for absorption. Moving on to the nucleus, and the nucleus is going to be formed of the genetic, it's actually going to be the genetic library with blueprints for nearly all cellular proteins. So inside the nucleus, I have the codes that will tell me how to form every single protein that can be produced in any of your cells. And the nucleus is going to be responding to signals and dictates kinds and amounts of proteins that would be formed. Most of the cells are going to have only one nucleus with few exceptions, like the skeletal muscles cells, for example, as we've seen back in the tissues, skeletal muscle cells were multinucleated. They didn't have more than one nucleus, but most likely every cell is going to have just one nucleus. So looking on here, this is my genetic material. The genetic material is condensed in a chromatin form. Surrounding this genetic material, we're going to have a double layered membrane. This is called my nuclear membrane. And you will see that the nuclear membrane has openings. Those are called my nuclear pores. You will see the outer membrane of the outer layer of the nuclear membrane is continuous with the rough endoplasmic reticulum. Because as we're going to learn, I need the codes on how to produce the protein to be delivered from the nucleus. And this code is going to be used on here by the ribosomes to produce the protein. Because remember, where do you have the blueprints on how to produce the protein? In the nucleus, in the DNA. And where do you actually produce the proteins? The proteins get produced in the ribosomes, by the ribosomes, in the rough endoplasmic reticulum. So they need to be connected to one another. Why? Because simply this one, the nucleus is going to be providing the code, and the ribosomes are going to be following this code to build up the proteins. So you're going to see that the outer layer of the nuclear membrane is going to be continuous with the tubules of the rough endoplasmic reticulum, as we've seen on here. At the center of the nucleus, we've got the nucleolus, which is the site for the formation of the ribosomal RNA, which enters in the structure of the ribosomes. So you see on here, the nucleolus is the site for the formation of the ribosomal RNA, which is actually going to enter in the structure of the ribosomes. So we're looking on here, first the nuclear membrane. It's a double membrane barrier that contains pores. The outer membrane is going to be continuous with the rough endoplasmic reticulum and bears ribosomes as well. Inner lining, or what we call the nuclear lamina, it's going to be maintaining the shape of the nucleus. The pore complex is going to be responsible to regulate the transport of large molecules in and out of the nucleus. See on here, double layered membrane. This is my nuclear membrane. I have the pores, which are going to be responsible for regulating what passes in and out of the nucleus. And I will see that the outer membrane of the nucleus is continuous with the cisternae of the rough endoplasmic reticulum. And the inner membrane is the one that would be maintaining the shape of the nucleus by containing the chromatin. The nucleolus is a dark staining spherical body that will be located within the nucleus. It's going to be serving as a site for the formation of the ribosomal RNA, which is required for the ribosome formation. And also it's going to be responsible for the ribosome subunit assembly. So it's going to be responsible, generally speaking, for the production and formation of the ribosomes. Moving on to a really important topic in this chapter, which is the cell cycle. What is the cell cycle? The cell cycle is the life cycle of a cell. If this is the life cycle of a cell, like this, the cell remains most of the time growing. And during this time, here, the cell is just growing. And for a limited time, the cell is going to be dividing. So we divide the cell cycle into two main phases. Two main phases that will be forming my cell cycle. The time during which the cell is going to be growing, this is my interphase. And the cell division phase is going to be the mitotic phase. If we're looking at the interphase, which the time during which the cell is going to be growing, we will see that interphase is going to be divided into three main phases, which are G1, growth 1, S, synthesis phase, and G2, growth 2. I'm looking on here. G1 is the time where I'm building up cytoplasm. I will be forming my organelles. I'm getting the organelles ready for cell division, because if the cell gonna be dividing, you're gonna need to double all of its contents before it performs the cell division. Because if you're gonna be dividing into two daughter cells which resemble to the mother cell, you're gonna need to have double everything that you've got inside the cell before the cell divides. During the S phase, you're going to be still performing growth of the cell, but in the same time, you're going to be doubling your genetic material. This is the time when the DNA is going to be formed. I will be replicating the genetic material during the S phase of the interphase. This takes us to G2, where the final preparations for the cell division are going to be taking place. After that, the cell starts the mitotic division. For this cell to become two daughter cells, which are identical and resemble the mother cell after completion of the mitotic phase. What if I'm dealing with a cell that does not divide? It's a permanent cell. If the cell is a permanent cell, It doesn't divide. Like what? Like the cardiac muscle cells, like the nerve cells, like skeletal muscle cells. Those are permanent cells. So would they be able to grow? Yes, they are able to grow. They remain forever in G0. So if you go to the gym, you are working out, what's going to happen to your muscles? Your muscles cells will grow. I will have hypertrophy of the muscle because those cells are capable to grow. But did you increase the number of muscle cells in your skeletal muscle? No, you did not. You did not increase the number of muscle cells. What you've done is an increase in the size, this is growth of the muscle cell. That's the same numbers that you already have. It won't change. Unlike the epithelium, for example, every day I'm shedding off the dead cell layer of my skin. I'm shedding off the keratin layer, which are the keratinized cytoplasmic membranes of the dead cells, and those get replaced. So the outer layer of your skin, for example, gets replaced every month. So here I need cells to proliferate. I need cell to divide to increase the number of cells. All right, so how the cell looks like during interphase. So if I'm drawing a cell on here, this is my cytoplasmic membrane. This is my nucleus inside. This dark center in the nucleus is my nucleolus. So I have the nucleolus, I have the DNA located inside the inner membrane of the nucleus in the form of condensed chromatin, like this. To the outside of the nuclear membrane, remember we've got those sets of tubules. Those is my, you see on here, the centrosome formed of two centrioles. So we've got the centrosome on here. This is how the cell gonna look like during what phase again? During interphase. So the organelles are gonna be dividing, I'm producing more organelles, I'm increasing the size of the cell by increasing the amount of cytoplasm. During the S phase of the interphase, you're gonna be duplicating the genetic material, so I'm doubling the chromatin inside the nucleus. So all this is gonna be taking place during interphase, which are actually three phases, G1, S, and G2. Because we figured out that the duplication of the DNA is taking place not all the time in a phase in the middle of the interphase. So I'm done duplicating the genetic material. I'm done increasing the size of the cell. I'm ready now to perform a mitotic division. I'm ready to perform a cell division. So this takes us to the first phase of the mitotic cell division. The first phase is going to be mitosis. What's mitosis? Mitosis is the nuclear division. So I'm dividing the nucleus into two nuclei. So what's going to happen here in order for you to divide the nucleus? First, the cell passes into a primary phase. This is my prophase. And during prophase, notice here what's gonna change compared to the interphase. This is again my cell like this. The centrosome is gonna split into two centrioles. Each one is gonna be traveling to one pole of the cell. Also, the chromatin here inside the nucleus is going to be condensed into chromosomes. And each chromosome is going to be formed of two sister chromatids attached in the middle by a centromere. So if I'm drawing here, this is my chromosome. It has two arms attached in the middle. Here like this. Those are the two sister chromatids, and they are attached here at the centromere. All right, so I've condensed the chromatin into chromosomes. This is my chromosome, which is the condensed form of the chromatin. This is early prophase. Also, what happened is here, the nuclear membrane was broken down, and the nucleolus here did disappear. All this takes place during early prophase. During late prophase, those centrioles of the centromere are forming mitotic spindle fibers. They will reach the two different poles of the cell, and those mitotic spindle fibers, those protein filaments, will be attaching the chromosomes at the point of attachment between the two sister chromatids. Here, I'm attaching the two chromosomes. I'm attaching the chromosomes at the point of attachment between the two sister chromatids forming the chromosome. This is my centromere. So the mitotic spindle fibers here will be attaching those chromosomes at the centromere. And they will start to pull those chromosomes to the two poles of the cell. So this takes us to our next phase of the mitosis, which is my metaphase. During the metaphase, as the mitotic spindle fibers keep pulling the chromosomes to the two poles two opposing poles of the cell, the chromosomes gonna be arranged at the equator of the cell. And the centromere is gonna be forming a line together. We call this line on here, formed by the arranged centromeres at the equator of the cell, we call this is my metaphase plate. Metaphase plate. As I keep pulling the two sister chromatids away from one another, I will have a rupture of my centromere and the two sister chromatids are going to be pulled away from one another, moving towards the two different poles of the cell. And as they are dragged in the middle, this is how would they would look like. They have this V-shaped appearance of the chromosome. So what happened here during anaphase is the rupture of the centromeres, the separation of the two sister chromatids forming the chromosomes, and now I have formed two sets of chromosomes that contains a complete genetic information of the cell. So how come do you say you get two sets of chromosomes, although you call this one here as a chromosome? Why you don't call them two sets of chromatids? All right, so the chromosome has two forms. Either, so imagine of a chromosome is like a book. That you have, and this book contains an information. The total information is what we call a chromosome. So again, what's the chromosome? It is the information in this book. So if I told you I have two copies of the same book, same exact book, did my knowledge change? What do you think? Did I add any information? Having two copies of the same book, does this mean I've got more information? What do you think? No. I still have the same amount of information as if I did have just one copy. So this is the same concept. So here, I did have two copies of the same book. So in terms of information, you still have one book. You still have one chromosome. But when you separate the two copies, so now each set has a copy. So each set has a book. Each set has a chromosome. A chromosome here is a complete set of information kept in the book. So if I separated the two from one another, yes, first I did have a chromosome formed of two copies of the information. So it's called chromosome. And we call each one, it's a copy of the book, chromatid. But once they get separated, now each one is going to be a book, a separate book. It's not a copy anymore because they got separated to two different sets. All right, so the phase during which this split of the centromere is going to be taking place and the formation of the two sets of the chromosomes going to be taking place, it's what we call the anaphase. Ana, like anatomy, is you cut, you split. All right, you dissect, it's going to be anatomy. Anaphase, you are splitting here the centromere of the chromosomes, separating the two sister chromatids into two different chromosomes. In the final phase of the mitosis, again, what's mitosis again? It's the nuclear division. It's the division of the nucleus into two nuclei. So now I've got centromere on here, a centrosome on here, and I've got sets of chromosomes. The nuclear membrane will reappear. The nucleolus will reappear, and the chromosomes are not going to be kept as chromosomes, but they will turn into the condensed form, into the chromatin. Of course, the mitotic spindle fibers will disappear and will not be attached anymore to the chromosomes. Again, nuclear membrane reappears. Nucleolus will reappear, and the chromosomes will form the chromatin on the inside of my nucleus. In the same time, so we call first this phase is my Tito phase. Tito phase. During late anaphase, you will notice that there will be a constriction ring that is appearing at the equator of the cell, separating the two sets of chromosomes that got formed. This constriction ring, it's going to be also taking place during Tito phase, while the two nuclei are getting formed, and they will finally separate the two nuclei from one another, forming two daughter cells, which will be identical to the mother cell and identical to one another. All right, so yeah. We got here's the two daughter cells. Each one has its own nucleus, the chromatin inside, nucleolus, and it has its centrosome and its cell organelles. What do we call the processing here, during which we're going to be splitting this cell into two daughter cells. The cell division is what we call cytokinesis. It's the cell division. One of the most common wrong answers that they see on the exam is people choosing mitosis as cell division. So mitosis is not the cell division. Mitosis is nuclear division. It's the division of the nucleus. Cell division is cytokinesis. So both, you see on here, both mitosis and cytokinesis are going to be part of the mitotic phase of the cell cycle. Both mitosis and cytokinesis are going to be part of my mitotic phase of the cell cycle. So again, what is the cell cycle? This is the life cycle of the cell. The cells that have the ability to divide again are going to be passing through an interphase, which consists of G1, S, and G2. And the mitotic phase, during which these cells are going to be dividing. So we're looking on here, mitotic phase, again, I have two parts of the mitotic phase. Remember, during interphase, cell on here did have nuclear membrane, did have a centrosome, and it had a nucleolus, and the chromatin is the form of the genetic material. So what happened during early prophase? See here, the chromosomes are going to be condensed and will be formed. You don't have chromatin. The two centrosomes are going to be separating, and they will be traveling to the two different poles, two opposing poles of the cell. And they are forming here the mitotic spindle fibers. During late prophase, you'll see that the nuclear membrane did break down. So I have broken down the nuclear membrane, and the mitotic spindle fibers will be attaching the chromosomes at the point of attachment of the two sister chromatids at the centromere. Mitotic spindle fibers are going to be pulling the chromosomes from both sides. So you're going to see that the centromere are going to be arranged at the equator of the cell, as you see on here, and we call this line that gets formed in at the equator of the cell is going to be my metaphase plate. As I keep dragging the two chromatids away from one another, this will allow me to split the attachment, the centromere, dragging the chromosomes to the two different poles of the cell. And you see as they are dragged at the center, so they will have this V-shaped appearance. I formed two sets of chromosomes. Each set of chromosomes is going to be located on one side of the cell. The nuclear membrane will start to reappear as part of the Tito Phase, the last phase of the mitosis. The mitotic spindle fibers will be broken down, and the chromosome will form the chromatin another time, and the nucleolus will reappear inside the newly formed nuclei. So now I've got two nuclei in the cell, which is forming a constriction ring that will be separating the two nuclei into two separate cells, into two daughter cells, and this step is what we call the cell division or cytokinesis. Questions? All right, for question number 3 for today's in-class activity, please. Because this is the third question of your in-class activity. So during what stage of the cell cycle is DNA replicated? When would you be replicating the DNA? What do you think? When would you have the replication of the genetic material? Any idea here? Waiting for some more responses. What do you think? Remember, the duplication of the genetic material is taking place when? During the S phase of the interphase. Don't be misled here. You're not duplicating the genetic material. Here, you're not duplicating the genetic material. You're just separating the two copies. But when did you make the other copy of the book? During interphase. Exactly. Interphase is going to be the time when the cell is going to be growing. And again, this is split into G1, S, and G2. The DNA is going to be duplicated during the S phase of the interphase. All right, so we can have a 10 minutes break. We come back to discuss a very important topic for us. It's going to be the protein formation, which includes the transcription and translation from the DNA to RNA to protein. All right, so we can have a 10 minutes break. We come back to have a brief discussion of the duplication of the DNA, transcription and translation to produce the protein. Any questions before we go for a break? Any questions before we go for a break? All right, so we're gonna have a break for 10, and we'll see you in 10 minutes. be responsible for the production of the proteins. So the genetic material kept in your nucleus is in the form of a DNA, and the DNA is going to be formed by building blocks. Those building blocks are called the nucleotides. Nucleotides are going to be formed of a pentose sugar. Penta means 5, so it's a 5-carbon sugar. Attached to which we've got a phosphate group on one side, and on the other side here, attached to the pentose sugar, we've got a base, a nitrogen containing base. So looking at those building blocks, the nucleotides, is going to be, they will be different from one another according to the type of base they contain. So we have 2 families that we might have attached to the pentose sugar in the DNA and RNA. It's going to be the same concept in the RNA with some differences, as we're going to see in a second. Those are either going to be purines or pyrimidines. Purines are like the adenine and the guanine. And the pyrimidines are going to be like the thymine and the cytosine. A major difference between DNA and RNA is that RNA doesn't have thymine. Instead, it has another pyrimidine. It's called uracil. DNA is present in a double strand form. So you're going to have 2 strands of nucleotides, and those nucleotides are going to be paired together according to the type of base each nucleotide going to contain. So let's say this is going to contain adenine, this is going to contain guanine, this one is going to be thymine, this one is going to be cytosine, guanine, adenine, cytosine, thymine, guanine, let's say. So in order for those bases to be paired together, adenine pairs with thymine, and the guanine is going to pair with cytosine. So can you tell me the sequence on here on the other strand of the DNA? So adenine pairs with thymine. So what's going to be the first base on the other strand? Thymine, exactly. About the next one, cytosine. Next is going to be thymine pairs with adenine. Cytosine pairs guanine, exactly. Can you keep going on here? Just tell me the bases on the other strand for the remaining part of this DNA molecule. So C, T, G, G, A, C. All right. So in order for me to form another copy of the DNA, I will need something called DNA polymerase. And we're going to have different types, but we just keep it simple at this point. Polymerase. What the DNA polymerase is going to be doing, it's going to be unwinding those strands. I'm separating the strands from one another, and it's going to be adding the base corresponding to each of those nucleotides. It's going to be adding new nucleotides to form a new strand for each of the two primary strands of the two parent strands. So for example, here, it's going to come to this strand. It's going to add a nucleotide containing T, C, A, G. So I'm using a strand, the original strand, as a template to build up my new strand. About this other strand, what would I do with it? I would use it as a template to build up my new strand. A. I will read and add the corresponding nucleotide containing the corresponding base. So it's going to be G, going to be T, and C. So what happened in here, by adding those nucleotides according to the bases on each of the two strands, using the two primary strands as templates, what I will end up with, I will end up having two copies of the DNA. So you see on here, I've got two double strands. I'm building up a new strand for each of the two parent strands. So I end up doubling or duplicating my genetic material. And this is what happens during the S phase of the interphase. This is the DNA replication. How would I be able to produce proteins? Remember, those codes on the DNA are coding for proteins to be formed. If I'm not producing another DNA, I don't want to double the genetic material. I'm not preparing for cell division, but I'm producing a protein, a hormone, whatever kind of protein that this cell is designed to produce. So what will I do? I will need another enzyme called the RNA polymerase. And the RNA polymerase will work in the same pattern like the DNA polymerase. It will come here and will be unwinding the two strands. I would be using just one strand this time to build up my RNA, because the RNA is a single-stranded molecule. It's not double-stranded like the DNA. It's a single-stranded. Remember that the RNA doesn't contain thymine. Instead, the adenine pairs with uracil. So can you give me here the RNA that gets formed? Because remember here, why would I need to form an RNA? Why I don't just copy the DNA? Remember, the DNA is the library. So it's gonna keep going like this. And I'm not interested in copying the whole library. I'm just interested in producing a single protein. So what will I do? I will look for the code that will tell me how to produce this protein and will copy just this code needed for the specific protein I'm interested in producing. If you don't want to get a copy of the whole library to get an information about a very small topic. So what will I do? I will go to the library, I will get a copy of the few pages I need, and I will get the information required for me to build up this RNA. The RNA that we're going to be producing is called the messenger RNA, and from its name, it serves as a messenger. It's just going to be delivering the code from the DNA, which is located inside the nucleus, to the outside of the nucleus where you've got the ribosomes. Remember, inside the nucleus, I've got my genetic material in the form of chromatin. This is my DNA. And to the outside, I've got those ribosomes, and those are the factories responsible for the production of the protein. So what happens in here? I go inside and read the specific codes of the proteins that I want to produce, and this is gonna be made by pairing the bases on the DNA. So I will be reading just one strand to get my messenger RNA, not the two strands. Say this is a code coding for the production of a protein called the myosin, let's say. Myosin is one of the proteins located in your skeletal muscles and cardiac muscles, which is going to be responsible to allow the muscle contraction to take place. So let's say I am now located in a skeletal muscle. I did find the code for the production of the myosin. What I'm trying to do now, I'm trying to copy the code. So what will I do? I would be forming a messenger RNA by adding one nucleotide at a time to form this strand of complementary bases. So for the adenine, what's going to be the first nucleotides that I would be adding to this messenger RNA in order for it to be formed? What do you think? Remember, in the DNA, adenine pairs with thymine, but we don't have thymine in the RNA. We have instead uracil. So what's going to be the nucleotides that we're going to be using? The nucleotide that contains uracil, so U. Can you tell me what's going to be the code on my messenger RNA? Can you complete the code on the messenger RNA here for me? Please add this as your in-class activity number, I believe 5 or 4. This is your in-class activity number 4. So please add the code on the messenger RNA that will be matching the nucleotides on this strand of the DNA. I will give you a few more seconds. All right, so adenine pairs with uracil, right? Then G pairs with C, T pairs with A, adenine. Do you have adenine in your RNA? Yes, I do have adenine in my RNA, so I don't have a problem. So it's going to be UCA, then C pairs with G, G pairs with C, A pairs with, do you have T, thymine, in the RNA? Do you have T? Yes or no? No, I have uracil, so A pairs with U. C, G, C, G, T, adenine, I have A, yes. Then G pairs with C, all right. So what's gonna be the sequence here? It's gonna be U, C, A, G, C, U, G, G, A, C. All right, so let's forget about the last one on here. So I've formed my messenger RNA. What do we call this procedure in which I produce a messenger RNA from a DNA? So this molecule needs to be modified and trimmed. We're gonna have some regions that will have a code and some regions are not coding for anything. So we're gonna get this messenger RNA trimmed, but just focus at the basics for now. All right, so this messenger RNA is produced according to the strand, the template strand on the DNA, and this is gonna be a procedure that we call the transcription. So again, what is transcription? Transcription is the formation of a messenger RNA from my DNA. This is what we call the transcription. Messenger RNA is now located inside the nucleus of the cell. I've got, this is my nucleus on here, and I've got this here as my messenger RNA. So again, what was the sequence on here? It's UGA, GCU, and GG. I want to use this sequence on the messenger RNA to produce my protein. So this messenger RNA is going to be leaving the nucleus through the nuclear pores. This is where it gets in contact with the ribosomes. This is just one ribosome here. I'm drawing. Ribosomes will have two subunits like this, a large subunit and a smaller subunit. And we'll have three main sites to accommodate, as we're going to see, the transfer RNA. So amino acids in your body, in your body that you are using that are actually present in nature. In order to produce a protein, you're gonna have to have access to 20 different types of amino acids. So in nature, we do have 20 different types of amino acids, and we're gonna be using all of them to produce our proteins. Let's say we're gonna give them numbers rather than names, all right? So you're not required to know the names for this course. If you're taking biochemistry later on, they will be teaching you the chemical differences between those amino acids, and you're gonna need to know the names of those 20 different types and their chemical characteristics, but not for us here in this course. All right, so we have 20 different types of amino acids, and on the messenger RNA, if you notice on here, you have those nucleotides attached to one another. The ribosome is going to be reading 3 bases at a time. Each 3 bases are going to be coding for 1 amino acid. So what is going to be the first codon on my messenger RNA is going to be UGA. This is a very bad example here of a code. So what do you think is, if you do have 20 different amino acids and each amino acid is going to be coded for by 3 bases. 3 bases are going to be coding for a single amino acid. So what do you think is, how many codes are going to be available for the amino acids? Or in other words, let me reword my question on here. So for each amino acid, would you have a single code or multiple codes? What do you think? If I will be using the combinations of those 3 bases. So what do you think? Just a guess. Will you have more combinations of the 3 bases or more amino acids? Many, right? I have multiple codes. Because you see on here, let me give you some examples. So if the first one was, the first base was A, for example. Second one was T. We don't have a T here. We have U, let's say, or A, another A. So it might be AAC, AAG, AAU, AAA. First one was A and the second one was U. So it might be AUC, AUG, AUU, AUA, and so on. So you're going to have a huge number of codons formed of combinations of those 3 bases on your RNA. So I'm expecting that each amino acid is going to be coded for by more than one codon. And you see on here, those are my amino acids, and those are the codons for each of them. So phenylalanine, for example, has 2 codons, UUU, UUC. Leucine is going to have 2 codons, UUA, UUG. Cysteine is going to be UGU, UGC. Arginine, it has 4 codons, CGU, CGC, CGA, CGG, and so on. So you see, each amino acid is going to have more than 1 codons that codes for it. So how's this codon going to be working? So when the ribosome starts to read the codon on the messenger RNA, it reads UGA. So let me use another one because UGA is a termination codon. You see on here, UGA is a stop codon. So if the ribosome reads UGA, it will stop the formation of the protein. So it cancels everything. So it's a very bad example on here to start with. UGA. So let's see, for example, I have AUG instead. So it reads AUG. It will start to call for another type of RNA called the transfer RNA. And the transfer RNA has 2 parts. A part that is attached to the amino acid. So, and a part that will be carrying the anti-codon. So AUG is not coding for an amino acid, as you see on here. It just tells the ribosome that this is where you're going to be starting the production of a protein. So nothing will come, no transfer RNA. So let's move on to our next code. The next codon is going to be GCU. So I will read the messenger, the ribosome is going to be moving along the length of the messenger RNA, reading the next code, which is going to be GCU. When I read GCU, I will start to look at this table, but of course it won't in reality. So we'll start to look on here, GCU, alanine. So this codes for alanine on here. So what happens? How would I call for alanine to come in? Alanine is going to be attached to a transfer RNA that carries the anti-codon. So what do you think is going to be the anti-codon that will fit with GCU? The anti-codon on here, on the transfer RNA. What do you think is going to be the complementary anti-codon? Is going to be CGA. So you're going to have transfer RNA that carries the anti-codon, also is going to be attached to the specific amino acids that I'm coding for. So what is the function of the ribosome in here? It's going to be reading the codon on the messenger RNA, and it will call for the transfer RNA to come in. As it comes in, and the anti-codon on the transfer RNA is going to be matching here the codon on the messenger RNA. It brings in the specific amino acids that this codon is coding for. And this one is my first amino acid here. It's going to be alanine. And then the ribosome is going to keep moving along the length of my messenger RNA. So it's going to read the next code. This transfer RNA here, still inside the ribosome, and the ribosome will start to read the next code. So it reads GGA. So according to the table on here, GGA is going to be coding for glycine. So the next amino acid that will be coding for is going to be attached to a transfer RNA. See on here, another transfer RNA in the cytoplasm that carries the anti-codon for GGA. So what do you think is going to be the anti-codon on the transfer RNA? CCU. And what amino acid would it be carried on this transfer RNA? Again, according to the table on here, it's going to be glycine. So you've got here glycine that will be attached to this transfer RNA. So the ribosome function is to call this transfer RNA to come in along with its amino acid. So transfer RNA will bring the amino acid. It will be matching the codon on here to the next codon, which is this one on here. It's going to be matched. And the two amino acids are going to be attached to one another. So you've got alanine first, and then you've got glycine. And this transfer RNA is going to be moving to this exit site on the ribosome, and it's going to leave the ribosome without the amino acid. So transfer RNA exits the ribosome, leaving the amino acid, and then I will read the next codon on the transfer RNA as I keep moving along the messenger RNA. I keep reading 3 bases at a time, coding for a different transfer RNA to bring in another amino acid, and another amino acid, and another amino acid, and so on. So what am I seeing on here? I will see that the ribosomes are going to be moving along the length of the messenger RNA, reading 3 bases at a time and coding for transfer RNAs that will be carrying the corresponding amino acids. And this will allow you to build up a chain of amino acids. And this chain of amino acids that you're building, it's going to be the polypeptide chain that will be forming the final shape of the protein. This chain of amino acids is actually the primary structure of the protein. So how come my nails, my hair, the pigment of my skin color, the enzymes, hormones, all of those are polypeptide chains like those, which are chains of amino acids? What makes them different from one another if they are all formed by chains of amino acids? Here comes the difference between the different protein is going to be determined according to the sequence of those amino acids. So for example, if I have a chain of amino acids like this, just a very, very, very basic example. And one of the amino acids on here has a negative charge, and the other one on this side has a positive charge. So what do you think is going to happen between those two amino acids? What do you think is going to happen between those two amino acids, attraction or repulsion? What do you think? If one amino acid carries a negative charge and the other one carries a positive charge. So what's going to happen between the two sides? Attraction. What if I told you a mutation happened? So rather than getting an amino acid on here, which is negatively charged, I got another one which is going to be positively charged. So this is positive, this is positive. Would this chain bend ever? Can I put this together? Can I put them together like it used to be? No. You see on here, this is how mutations are taking place. You change the amino acid, and as you change the amino acid, what's going to happen? If it had different properties, it will cause a disturbance in the shape of the protein. So the shape of the protein is going to be determined according to the sequence. So for example, I have part here that is hydrophobic, so it's going to be moving inside. That is hydrophilic, is going to be moving outside, and so on. So you have changes in the shape as if you are, have you ever seen this 3D, the 3D pen? So that uses plastic to draw in 3D. So what is actually coming out? It's a plastic band that's like, and what you're going to be doing, you're going to be moving around, you're building a 3D model using this pen. All right. So this is the same exact concept. So I use the chain to build up whatever I want. So some will build up a castle. Others will build up a box. Others will build up a rabbit, or whatever you're going to be drawing here with this pen. So it's the same exact concept. So you are modifying the shape, but here's the difference, is that you are not modifying. So it's not all neutral. The chain is not neutral like what you are using to draw on here, but each part has its own properties, its own chemical properties, and it's going to be drawing its 3D shape on its own. So you don't have to use it to draw. Actually, it's going to be drawing itself, giving you the final structure of a protein. So for example, a protein that is acting as a channel to allow sodium. A protein that will act as a carrier, like the sodium potassium ATPase pump. It has 2 binding sites for potassium, it has 3 binding sites for sodium. It can attach to an ATP. All this is simply a chain of amino acids that are producing this 3D final shape of the protein according to what again? According to the sequence of amino acids. And what determines the sequence of amino acids? What will tell you is that this first one is alanine, the next one is glycine, the next one is arginine. What will tell me the sequence of those amino acids by which I am building the chain is actually the sequence of codons on my messenger RNA. And what did determine the sequence of those codons on the messenger RNA is actually the sequence of the nucleotides in the DNA. Because what happened in here? I did read the template strand on the DNA to produce a messenger RNA, transcription. And then I use the messenger RNA to produce the protein. This is what we call the translation. Both together is called gene expression. So I'm expressing the gene. So I read the gene that codes for a protein, a specific protein on my DNA, and accordingly, I will be producing this protein. This is called gene expression. All right. Any questions? Any questions? All right, so this completes our discussion for today. I will be adding in an announcement on Canvas a really interesting video animation of the transcription and translation. I highly recommend you to go ahead and watch it. Yeah, so we've got only 4 questions for today, yes. All right. So again, a reminder, next week we're gonna, your lab exam and lecture exam are gonna be available. For 1 week to end on their due date, their scheduled due date. So again, expect the exams to be available 1 week before the due date. And remember that the best source for you to study for the tissue lab exam, which is going to be out of 100 points, remember, is by completing the visible body assignment. This is one thing. Also to get a good impression about how the question is going to look like. If you go to Canvas, you open lab exams review documents, you're going to see here tissue sample questions. Those are just simple questions for you to get an idea about what you're going to be tested on in this part. For chapter 2 is only going to be tested as part of the lecture exam. It's not going to be in the lab exam. So chapter 2, I will be posting a video from another professor that covers chapter 2. So let me look up in here and I will check one just to double check if the lecture exam gonna have the different chapters. All right, so let me check this out in a second. So we're looking here. All right, so what I will be doing for chapter 2, I will be posting a video recording from another professor who covers chapter 2. It's going to be matching our PowerPoint, so you don't have to worry about different information. The one thing. The other thing that chapter 2 information is going to be needed only to complete the quiz. It's not even going to be covered in the lecture exam. So chapter 2 is not going to be covered in the lecture exam. So looking here, we got lab exam 1. This covers only tissues. The parts that we mentioned that are covered in the tissues lab exam, which are part of chapter 4. For lecture exam 1, and again, you're going to be using these sample questions on visible on Canvas, and very important also to get the lab assignment on visible body courseware completed for you to practice the tissues lab exam. Also, next class I will be doing a quick review of the tissues. All right, I will be doing a quick review. So get prepared to have a quiz live with me next class covering the tissues. All right, this is just for you to become ready and to practice for the upcoming lab exam. For the lecture exam, it's not going to be covering chapter 2 anymore. It's going to be covering chapters 1, 3, and 4, which are the 3 chapters that we did discuss together in our live meetings and the recorded session. So for chapter 2, you're gonna need to have an idea about the chemistry. So I will be posting a video recording for it by the end of the week. That covers chapter 2, and you're gonna need this information for you to complete the chapter 2 quiz. Chapter 2 is simple if you've got any chemistry background. All right, any chemistry background is going to be working fine. Also, I will try my best to record my own video, but if I didn't get enough time to record my own, I will be posting another one that fits our curriculum. Any other questions? So again, lab exam and lecture exam are going to be available for you to complete 1 week before the due date. Modification here, we will not be tested on chapter 2 in the lecture exam. Is going to only cover chapter 1, 3, and 4. Video recording for chapter 2 is going to be posted either for me or from a different professor. And this will serve you to complete chapter 2 quiz. All right, so this completes our discussion for today. Please leave your last name before you leave the meeting. Thank you so much and have a good rest of your day.